Bismillahirrahmanirrahim. A very dear listener, in today's session we will talk about the human red blood cell or erythrocyte. Our today objective will be what is red blood cell, what are the normal shape of red blood cell, and what is the structure of red blood cells, and what are the composition of red blood cells, what are the normal value of red blood cell, and we also cover up what are the properties of red blood cell and function of red blood cell, and after lifespan about 100 to 120 days what happened with red blood cell. Some we will talk that some of the components of red blood cell are reused and some are excreted from the body. So let's start. What is red blood cell? Our today objective is what is red blood cell, what is RBCs, number second, what is the normal shape of RBCs, and third objective is what are the composition of red blood cells, and what made the structure of RBCs. We will talk about function of red blood cell. We will also cover up the objective of properties of RBCs, and what is fate of red blood cells. So dear listener, these are our objective which will be covered in today's session. That define red blood cell, or what are red blood cell, what is the normal shape of red blood cell, what are the normal value of red blood cell in human body, what are the components of composition of red blood cell, and what made the structure of red blood cell. We will talk in detail in this session about the structure of red blood cell, what are the functions performed by red blood cell in human body, what are the properties of red blood cell, and in the end what is the fate of red blood cell, or what happened with red blood cell after about 100 to 120 days. So let's start from the first objective. We will cover in this session all these objectives about red blood cell. Red blood cells, to remember what is red blood cell or define what is red blood cell, we have to remember some of words. For example, red blood cell is a anucleated cell, so we have to remember words anucleated. It's mean that red blood cell do not have any nucleus. Mature erythrocyte or red blood cell do not have nucleus, so red blood cell is anucleated cell and amitochondric. Number second point we have to remember is amitochondric, that there is no mitochondria present in the mature red blood cells. Okay, then it's contain hemoglobin, and the hemoglobin molecule which is present in the red blood cell is faked in a stroma. For example, this is a Hb molecule, this is a hemoglobin or Hb molecule in the red blood cell. It is faked in a stroma, and it has a specialized advantages that if due to any reason the red blood cell is cut up, so the hemoglobin does not easily or cannot easily escape from the red blood cell. So it contain hemoglobin, red blood cell having lots amount of hemoglobin which is present in a stroma. Then this is the introduction of red blood cell. That what is red blood cell? Red blood cell is a nucleated and a mitochondrial cell which is present in the blood. They are circulating and contain hemoglobin. And what is hemoglobin? We will talk in the next session about the hemoglobin. That hemoglobin is a specialized protein which is oxygen carrying protein present in the red blood cell. And biconcave shape and disc-like structure. Another point we have to remember about red blood cell, that is the size. The average diameter of red blood cell is 7.5 micrometer. So now we can easily define what is red blood cell. Red blood cell is also called erythrocyte. It is a nucleated and mitochondrial cell. It contains lots amount of hemoglobin which is interwoven in a stroma. That its advantages is that if due to any reason red blood cell is cut up, so the hemoglobin protein or hemoglobin fragment which is present in the red blood cell cannot escape easily from the red blood cell. It is a biconcave structure and disc-like structure, and the average diameter of red blood cell is 7.5 micrometer. This is our first objective, that how to define red blood cell. Now the second objective is what is the normal shape of red blood cell. So the normal shape of red blood cell is disc-like structure, biconcave structure, and the red blood cell is thinner in the center and thickest in the periphery or peripheral side. So this is the normal shape of red blood cell. Yet our second objective is what is the normal shape of red blood cell. So red blood cell, the normal shape of red blood cell is a disc-like structure and biconcave structure, and it is thinner in the center and thickest in the periphery. These disc-like structure having both functional and mechanical disadvantages. So let's talk about what is the functional disadvantages and what is the functional advantages of red blood cell disc-like structure, and what is the mechanical advantages of disc-like or biconcave like structure. For example, I am drawing a red blood cell over here. For example, this is a red blood cell. It is in the center, it is thinner, and in the periphery it is thickest, and it is a disc-like structure or biconcave structure. So this structure increase the surface membrane or permeability to diffuse much amount of gases through in, And another, this is the functional advantages or physiological advantages of red blood cell, that it increase the cell the surface area to diffuse much amount of gases inside the red blood cell. And the mechanical advantages of this biconcave or disc-like structure is that if the red blood cell pass through the capillaries smaller than red blood cell, so the red blood cell squeeze and pass through these smaller capillaries, and afterward it resume as original shape. This is the mechanical advantages of red blood cell. So our second objective is also cover that the normal, what is the normal shape of red blood cell. The normal shape of red blood cell is RBC or erythrocyte, is a disc like structure, and it is biconcave. It is thinner in the center and thickest in the periphery. And this disc like structure or biconcave structure having both functional and mechanical advantages. Functional advantages is that it diffuse much amount of gases, and this biconcave or disc like structure has mechanical advantages, which is squeezing ability. It can squeeze through the capillaries membrane smaller than red blood cell. Our third objective is what are the composition of red blood cell. So what is the composition of red blood cell? Red blood cells composition. So about 60 to 63% red blood cell consist of water, and about 37 to 40% RBC consist of solid material. In these solid material, about 34% out of 40, about 34% is hemoglobin protein or hemoglobin pigment, and the remaining is other protein. So I am showing in a diagram, I chart the composition of red blood cell. So water about 60 to 63%, and solid materials is about 37 to 40%. In these 37 and 40%, 34% is HB molecule, and the remaining 3 to 6% remaining 3%, 3 to 6% is other protein or other material. Very simple. Red blood cell consists 60 to 63% of water and 37 to 40% solid material. In solid material, 34% is hemoglobin and 3 to 6% other material. What are these other material? These other material are, number first, ATP, some of oxidative enzyme, some of coenzyme, and some of glycoprotein. This is the composition of red blood cell. Again listen, red blood cell composition about 60 to 63% consists of water, and remaining 37 to 40% is consist of solid material. In solid material, 34% is hemoglobin, and remaining 3 to 6% is what other material. And other material what consists? It consists phosphate, ATPs, oxidative enzyme, coenzyme, and glycoprotein. This is the composition of red blood cell. Now we talk about the structure of red blood cells. what is the microstructure of red blood cell. So the third objective is structure of red blood cells. The structure of red blood cell is, as we know, this is a cell, and a cell is always bounded by a double membranous membrane layer which is made of phospholipid and glycoprotein. Same like here, the cell membrane of the red blood cell is a double layer, a bilayer membrane which is made of phospholipid and glycoprotein embedded in them. So let me draw red blood cell. This is a red blood cells. It is disc-like structure, thinner in the center and in the periphery it is thickest. This is the normal shape of red blood cell. Now from outer surface we start about, for example, this is another layer of around the red blood cell. So we can easily define that red blood cell is covered by a plasma membrane which is made of phospholipid and glycoprotein. In the cell membrane, on the outer surface of cell membrane of the red blood cell, there is specialized oligosaccharide present which determine the ABO blood grouping system. For example, this is the chain of oligosaccharide molecule. This is the chain of oligosaccharide molecule which is present on the surface of or on the plasma membrane of the red blood cell, which determine what? ABO blood grouping system. ABO blood grouping system. And the red blood cell membrane having different types of channels present on the surface membrane, different transporter present on the surface of cell membrane of the red blood cell. For example, this is a transporter present on the cell membrane of red blood cell, and sub-specialized ion channels. It is ion channels which is present on the cell membrane. For example, red blood cell having a transporter for glucose which is called GLUT1. GLUT1 means glucose transporter number 1, which is present on the cell membrane of the red blood cell. And ion channels such as sodium and ion channel for sodium and potassium, and also having another channel such as REG1. For example, I am drawing over here another channels, and this channel is called REG1, means RH associated glycoprotein. Some of the other transporter present in the cell membrane of the red blood cell for water, which is called aquaporin. For example, this is another transporter present on the cell membrane of red blood cell, and it is for what purpose? For water transportation. So it is called aquaporin protein or transporter. We transport water, and we mean aqua means water. And we know that there is no nucleus, no mitochondria. So the cytoskeleton, the structure, the main skeleton of the red blood cell is made up by different types of protein. For example, this is a protein which is called spectrin protein. What? spectrin protein which is present in the red blood cell. Another structure protein is, I am drawing here, that is actin. Actin is another types of protein, a structure protein which is present in the red blood cell. Another types of protein which is called integrin, which is present in the red blood cell. For example, this is integrin protein. This is what? This is integrin protein. I am showing in a black, and in a blue this is actin. This is actin protein. This is what? Actin protein. This is actin protein, and here I am drawing it spectrin protein in a red. This is what? Spectrin protein, which made of the cytoskeleton of red blood cell. There is another specialized types of protein, that is glycoporin. What? Glycoporin. This is also present in the red blood cell and made up the skeleton or the body of the red blood cell. For example, this is the glycoporin protein which is present in the red blood cell. In most important fragment which is present, I am doing it from star, that is hemoglobin. That is what? Hemoglobin. About 34%, it is 60% it is composed of water, and remaining is protein, and the most abundant components in the red blood cell after water is hemoglobin pigment. That is hemoglobin pigment which is present throughout the red blood cells. In these, up to now we learn about the internal or integrated protein of the red blood cell, that is spectrin, glycoporin, integrin and actin. In these four types of protein, the most important is spectrin protein, and spectrin protein is responsible to maintain the integrity and stability in the shape of red blood cell. If due to any gene mutation or any condition, if the gene or mutant which is responsible to make spectrin protein, and the spectrin protein is not made up in a very high quantity, so the person develop a genetic disorder called spirocytosis, a condition in which there is the genes which is responsible for the synthesis or production of spectrin protein is muted. So the person develops a disease called genetic spirocytosis. What happens if the spectrin protein is less in the red blood cell, then the shape of the red blood cell is not maintained and integrity and stability is not maintained, so the membrane become fragile and it's easily damaged by and easily destroyed by the splenic capillaries or the body immune system. So this is the structure of red blood cell. That red blood cell is a bilayer membrane, and on the red blood cell membrane there is specialized ion channel, transporter, gate and pump are present for different purpose. On the red blood cell there is a chain of oligosaccharide present which determine the ABO blood grouping system. In the red blood cell there is no nucleus, no mitochondria. They use glucose. The red blood cell utilize glucose for their energy. And other structural protein are supporting our cytoskeleton protein which make the cytoskeleton or blood cell. That protein is spectrin, glycoporin, integrin and actin, and the remaining parts is hemoglobin or HB molecule. Now what are the function of red blood cell? Our next objective is what are the function of red blood cell. So as we know, red blood cell perform three main function. What are these functions? Number first, it transport oxygen from the lungs to the tissue. Number second function of red blood cell is it transport carbon dioxide from tissue to lungs, and it determine the ABO blood grouping system. These are the main three functions performed by the red blood cell. Number first one is function. What are the function of red blood cells? Function of RBC is number first O2 transporter, O2 transportation. The hemoglobin molecule of the red blood cell combine with oxygen and make oxyhemoglobin. So simply we call it transport oxygen from the lungs to the tissue. Number second is CO2 transportation. This is Hb with combine with O2 to make oxyhemoglobin. And CO2 transportation, when hemoglobin combine with CO2, it make carbamino hemoglobin. So we can understand that the function of red blood cell is the transportation of O2 and from the lungs to the tissue, and removing or transportation of CO2 from the tissue to the lung. And the third function is the most important function is determine blood grouping, determine blood grouping system. This is our next objective. Let's talk about the properties of red blood cell. That what are the properties of red blood cells? So there are three main properties of red blood cell. Number first one is Rolex formation. Number 10, suspension stability, and the third one is the specific gravity of red blood cell. So the first one is this is what properties of red blood cell. Rolex formation. What is Rolex formation? Whenever the blood is escape from the vascular system, the red blood cell become a pile of coin. This is called Rolex formation, and this is the property of red blood cell. The second property of red blood cell is suspension stability. What does it mean? It's mean that whenever the red blood cell is circulating with other blood cell in the vascular system, so the red blood cell disperse or suspended in the vascular system. For example, let me draw. This is a capillaries, and this is white blood cell. I am drawing from blue marker, and this is from black. I am drawing thrombocyte, means platelets. It is platelets. And let me draw red blood cell. This is red blood cell because red blood cell is suspended throughout its present. This is suspended red blood cell. In between present, near to the basement membrane it's present, and throughout is present. This is the suspension stability of red blood cell. And what is the specific gravity of red blood cell? So the specific gravity of red blood cell is specific gravity of red blood cell is 1.1922 to 1.101. This is the specific gravity of red blood cell. At the end, we talk about the fate of red blood cell. That what is the fate of red blood cells? So the fate of red blood cell after 120 days, the red blood cell in the spleen destroyed and divided into different particles. For example, this is a senile RBC. In the spleen, the spleen is destroyed, and the component after destruction is iron, second is globin, third is 4-pyrin. So iron and globin are reused by the body, and pyrin are excreted either bilirubin or uroglobulin. In the form of bilirubin in the stool and in the urine is uroglobulin is excreted from the body. There are some factors which influencing the production of red blood cell, that is erythropoietin, a protein which is synthesized mainly by the kidney, about 90%, and 10 to 15% in the liver. Erythropoietin is influencing the rate of production of red blood cell. Male sex hormone also influencing the production of red blood cells. High altitude also, and some of the growth factor and vitamin also influencing the red blood cell. And let me repeat the today's session very briefly. That in today's session we learn about what is red blood cell. So red blood cell is anucleated mitochondria and biconcave disc-like structure. We having 7.5 micrometer and contain lots amount of hemoglobin which is interwoven in a stroma. Then we talk about what is the normal shape of red blood cell. So the normal shape of red blood cell is a disc-like structure and biconcave, thicker in the center and thickest in the periphery. And the functional and mechanical disadvantages of these biconcave structure and disc-like structure is it diffuses lots amount of gases and can squeeze easily, and afterward it's remain its shape. This is the mechanical disadvantages. Then we talk about the composition of red blood cells. So the composition of red blood cell is about 60 to 63% is composed of water, and remaining 37 to 40% is composed of solid material. In solid material 34% is hemoglobin, and remaining 3 to 6% is other material like different oxidative enzyme, ATPase, phosphate and coenzyme are present. Then we talk about the function of the structure of red blood cell. So the structure of red blood cell, it is covered by a bilayer membrane which is made of phospholipid and glycoprotein. On the cell membrane of red blood cells, some channel pump and gate are present for their specialized function. The transporter also present in the red blood cells, such as GLUT2 and aquaporin transport. GLUT2 for glucose and aquaporin for what? Water transportation. Ion channels such as sodium potassium pump are present. And on the cell membrane of red blood cells, some chain of oligosaccharides are present which is determine the ABO blood grouping system. Inside the red blood cell there is no nucleus, no mitochondria in mature erythrocyte or red blood cell, but the cytoskeleton is made up by spectin and crane glycoporin and crane protein which make the cytoskeleton. But the most important protein which have to remember is spectin, because it maintains the stability and integrity of the red blood cell. Then we talk about the function of red blood cell. So very easy, we have to remember three main function of red blood cell, that is O2 transportation, CO2 transportation and ABO blood group determination. Then we talk about the properties of red blood cell. So the property of red blood cell is Rolex formation. What is Rolex formation? Whenever the blood is drawn or the RBC is escape from the vascular system, it's become a file coin like structure on each other. This is what Rolex formation. Suspension stability is the second property of red blood cell. Versus suspension stability. Suspension stability, that the red blood cell in the vascular system or in the blood with other cell, it is suspended. not combined, moving or circulating to the body. It is dispersed in the vascular system or capillaries and arteries. Then we talk about the specific gravity, which is 1.192 to up to 1.1101. And at the last we talk about the fate of red blood cell or the lifespan of red blood cell. So about 100 to 120 days. After 120 days, the red blood cell move into spleen. It is destroyed by the splenic capillaries by different macrophages. We will talk later. And the components is iron and globin, which is reused by the body, and the waste product is porphyrin, and porphyrin is secreted either by bilirubin in the stool and uroglobulin in the urine. Thank you.